Okay, I'm Alexander, and this video is for my first essay for our first section in physiology. There's 3 points I wanted to talk about that are homeostasis, which is a point of preference that every system in our body has to where it stays happy and healthy, and the different systems that we have within our body are all trying to stay within this range that it prefers to be in. I'm also going to talk about how we transport things in and out of ourselves, because that's a lot to do with homeostasis. So we'll talk about that. And I'm also going to talk about osmolarity and tonicity when it comes to the transport in and out of cells. So to begin, homeostasis. Every system within our body has a range that it prefers to be in to function properly so that we could be healthy. There's a term we use, which is equilibrium, and that is this state that I'm talking about. It gets tricky because every system in our body, it has to work together, and we have to share this space that it has with every other aspect of our bodies. And so it gets kind of difficult when one system needs something and the other could need something completely different, but it works together. And that's the state of equilibrium or homeostasis that we're talking about. This is done by mainly feedback loops. There's a ton of things that go into keeping our body in harmony, but something I'm going to hit is mainly feedback loops, and mainly done by something called a control center. And this control center, most of them are in our brain. This is the decision maker, and so I want to talk about the process of when our body receives a signal that tells us to adjust and how our body does that accordingly. So I want to start with a couple of terms, and so a stimulus, this is a change in a variable that sends a signal to our brain. So it's prompting a change, and it's done by having something called receptors. Receptors are all in our body. There's a ton, every system has them. And so a big example I'm going to be talking about today is a change in temperature. And so if I refer to receptors as in our skin, that's what I'm talking about. So a stimulus, this could be, for my example, this is going to be a change in temperature. So if you're outside and say it drops 10 degrees, so it goes from, here in Rexburg, it's pretty cold. So say it goes from 40 degrees in the afternoon and it drops to 25 at night. What's going to happen is that's the stimulus. We have receptors in our skin that recept that information, that take it and is going to send it to something called a control center, which in this instance is located in the brain. So what happens is there's a change in temperature, it's a stimulus. Our receptor receives that information, and there's two terms, there's two pathways. One is called the afferent pathway, and this is referring to the pathway going from the signal, from the receptor to our brain. After that signal travels from the afferent pathway up to our control center in the brain's gonna take in what information was just received, and it's going to decide how to correct the deviation within what our body prefers. And once it does that, that signal is going to travel from the efferent pathway, which is the pathway going from the control center back down to what we're going to call an effector. That's easy to remember because efferent and effector, I think it helps me remember that because they sound similar. But the effector is going to be the means by which our bodies can correct the change in deviation from what we prefer. And so our body temperature has a preference to stay at, I think 97.8, around there. And what's going to happen is this effector, which in this situation, it could be our skeletal muscles. In temperature, we also, if it gets too hot, we sweat. So sweating those glands, that can be an effector. But for this instant, because the temperature is dropping outside, our body's going to want to warm it up. And so an effector in this situation would be skeletal muscle, which would cause shivering and an increase in metabolism. Those two things are going to try to knock our body temperature back up. And our body will draw blood from the surface of our skin back down deep within our bodies so that we can try to maintain the warmth that the crucial structures in our body need. That's how our bodies would receive a signal and how it would respond accordingly. So going off of that, something very important within this whole system is going to be the idea of a feedback loop. And so I'm going to erase the board. I'm going to draw something up on the board, and then I will continue the video and we can go from there. Okay, so we're back. I've drawn some things on the board to explain feedback loops. This one, this is going to be a negative feedback loop. I'll explain what that means. After that, I'll erase and I'll draw up a new situation, which will talk about a positive feedback loop, because they're really different, but you'll see they kind of go off of each other. So, okay, in this situation, talking about body temperature. Some important terms I'm going to be saying. I'm going to say a set point, and then here's feedback, and that includes the negative and positive aspects of a feedback loop. So feedback loop, kind of like the picture I had drawn up before. There's this system from a stimulus, your receptors that detect the stimulus, the afferent pathway, which goes from the receptor to the control center, back down the efferent pathway to an effector, which will ultimately correct a deviation according to what that system needs. So in this situation, that's going to be happening constantly. And that's what this loop refers to, is that pathway from stimulus to effector and back again. And that's why it's called a loop, because we're going to repeat and repeat until homeostasis or equilibrium is gained again. And so here, correction from my last video is I think I said the body temperature is like 97.5. Looked it up, 98.6. That's what we prefer to be at. So here I drew. Here's time, and then it's our body temperature. Goes from 96 to 100, just for an example. Here's the value, or this range, that our body wants to stay in, and that is around 98.6. And what this is going to be called, this right here, that is the set point, which is the point that that system prefers to be at to perform at its best. So we have this set point here. And when temperature drops or when temperature raises, this is happening constantly, depending on how many clothes you're wearing, the rooms you go, different classrooms, walking inside, outside. So this is constantly going. And what's happening is let's say it's too cold. So what's going to happen is those receptors sensing that, telling it to the control center and back down to the effector. I talked about previously shivering, the increase in metabolism, and blood movement from the maybe the outside of your skin more inwards, and that's going to try to warm you back up. So what's happening here is this is what's occurring right here in this blue line. Is body's temperature is a little too low, so those effectors are going to push it back up because it wants to be within this set point. And what happens if it gets too hot? So we sweat, and there's also blood movement, so we're going to push blood from the inside more to the outside of the skin, which will try to help us sweat more, burn off some heat. So what's happening is our bodies are constantly in this movement of going from cold to high, and what's happening is that loop that I talked about, this homeostatic control system, is going to be moving and doing these things, maybe not to an extreme extent, but they're going to be doing these things constantly to keep us within that set point. And this is what we refer to as a negative feedback loop. So that's what this would be. And the term negative, I'm just going to say feedback loop. When we think negative, we're thinking something bad is happening, or that, it's going ulterior to what it should be. But in this situation, we refer to it as negative because, let's say, as the system is going down, so as temperature is going down, our bodies are going to react negative to what it's receiving. So if temperature is going down, then our bodies are going to push it back up. And if it's going too high, then our bodies are going to push it back down. So that's what this refers to as negative, is depending on what is happening, this feedback loop is going to be opposite, and that's what the term negative refers to. And this is crucial. So when you hear negative, it's not bad. This is actually a really good thing that's happening, because this is what's allowing our bodies to function the way that we need to. Excuse me. Okay. So, yeah, we talked about set point. We talked about this negative feedback loop. Now, an important question is, what does the positive feedback loop do, and why is it necessary? Before I draw the example on the board, I want to talk a little bit about it. The positive feedback loop oftentimes, not always, occurs when there's something called an inadequate response, or this negative feedback loop. The changes that it's trying to make are inadequate for what's happening to our body. And that, most times, is where the positive feedback loop is going to come in. An important question to think about is why would we ever need something that's going to push us out of our set point? Because that's what a positive feedback loop is saying. Similar to this example where it's flipped, so what's happening, the opposite is going to occur through the effectors to get us back to where we want. In a positive feedback loop, instead of the response being opposite to what we're receiving, if the response is going down, or if the whatever happening to our body is going down, what's going to happen is the negative feedback loop is going to kick in. But if it can't get us to where we need to be, a positive feedback loop will come in and it will continue to push us down. That's why it's called positive, because what's happening, the response is going to be similar to that. because this negative feedback loop isn't getting us to where we want to be. So if something's going down, our response will also be to continue pushing it down. And same if it's going up, the response will be up. So I'm going to erase this, and I'm going to bring up an example of a time that is going to be important for this positive feedback loop to kick in our bodies. Well, of a positive feedback loop. Not the best artist. This is a brain. This is our, spinal cord. And this is the inside of the uterus when there's a baby. I did the best I could. And I drew stretch receptors on the outside of the uterus. And so this process that we're looking at here is going to be labor. And the positive feedback loop is crucial when we're talking about a woman going into labor. And so, yeah, basically, when we're going in, or when a woman is going into labor, what's happening is the uterus is starting to stretch because the baby's ready to come out. And as those stretch receptors feel the stimulus in this instance, the stimulus is, stretching. And what's happening is it's receiving that signal that it's stretching, and through the afferent pathway, it's going to go up to our brain. This right here is your pituitary gland, releases a lot of hormones. And in this case, a very important hormone for not just birthing, but for a mother-child relationship is oxytocin. And this hormone is going to trigger more contractions. And so instead of the receptors receiving that stimulus of stretch and going to the control center, and the control center coming back to mute the contractions, to prevent that from happening, the body doesn't want that. The body knows that it's time to begin pushing. And so it goes from the receptors to the control center, and this pituitary gland, as it keeps receiving signals that the uterus is stretching, it's going to pump out oxytocin. And oxytocin is going to come from your pituitary gland and travel not down the spinal cord, but through your blood. And this blood is going to reach back to the uterus with that hormone. And as that hormone continues to send signals to the uterus, it's going to promote more contractions. And so as this process continues to go on, the arrow example I used, as contractions increase, the production of oxytocin is going to increase as well. And this is what we refer to as the positive feedback loop. And it loops because, again, as it sends signals to the brain, the control center, it's going to continue doing this until the baby is delivered. And this is a huge reason why oxytocin is very important. They can administer it to mothers when there's not enough of that going on. So there is medical intervention in a situation where a mother wouldn't be able to produce as much oxytocin, or a lower amount. But this is what is very important about the idea of positive feedback loops, because if our body only works off of negative feedback loops, trying to keep us within a range, our body doesn't always need to be within that range. Because what if a medical emergency like this occurs and the solution is only going through, it's not preventing it from happening? So there's many instances where positive feedback loops are very important. But this is a very common one. A lot of people talk about it, mainly because it's so crucial to a woman's health during birth. So I hope this helps. This is the idea of a positive feedback loop. Next, I'm going to talk about transport in and out of our cells, what that looks like. And so, yeah, I'll erase this and I'll continue going on after that. Okay, so there's going to be 4 different types of diffusion or transport across a cell membrane. First, we're going to break it down into parts. It's really important to understand the concept of osmosis and concentration before we start. So I have written up here, I guess it's a typical diagram you'll see of ICF and ECF. ICF is the intracellular fluid, the fluid inside the cell, and ECF is your extracellular fluid outside of the cell. So this is important. The way we move things from ICF and ECF is through something called aquaporins, and this is crucial for osmosis to take place. Because this membrane that separates the ICF and ECF is very selective, and it's only permeable, which means certain things can cross through it. And so we need to understand that when we're talking about osmosis, we're only referring to water flowing from inside or outside the cell. That's important because there's going to be osmosis, and then we're also going to have concentration gradients. But yeah, it's crucial to remember osmosis is referring to the movement of water. Driving force, this is something that the simple idea is that when we have a certain concentration of one thing on one side and there's another concentration on the other side, that idea of homeostasis or equilibrium that we talked about applies to ions and chemicals inside this system. So there's something called osmotic pressure gradients. This is referring to the pressure gradient that is created from osmosis, the movement of water, and it occurs through these aquaporins. That's a simple idea. The first 2 types of diffusion we're going to talk about is simple diffusion and facilitated diffusion. It's important to understand osmosis and this idea of this membrane when it comes to diffusion and transport. So that's why we started there. So now this idea of simple diffusion is purely carried out by attraction and by kinetic forces. There's no protein needed. ATP or energy is not required. This is something that just occurs based off of concentration. Here I drew an example. One side has more of this concentration, it can be whatever you want, and this other side has a low. And what's going to happen is all of this on this left side is so bunched up, and it sees that on this other side there's more room. And what's going to want to happen is it's going to go from this high area to this low until there's an even amount on both sides. This occurs through a selective permeable membrane. Selectivity is very important to realize. This membrane that separates these 2 sides is only accepting of certain ions or chemicals. and nothing else. So it's very important. But yeah, this idea of simple diffusion is just going from an area of high concentration to a low. There's no energy needed. There's no protein needed to help move it. It naturally happens. And that's about it for simple diffusion. Next, kind of a lot of information. It's pretty, I try to make it as simple as possible. Facilitated diffusion, hence the name, is facilitated by more than just concentration gradients. The things that are facilitated tend to be bigger and polar compared to simple diffusion. Simple diffusion works best with smaller things and with non-polar things, although it can occur with polar. But when things are bigger compared to the simple diffusion and more polar, we tend to see a different type of diffusion. And so what's important to realize is in facilitated diffusion, we're going to have gated channels, and we're going to have channels that are mediated by proteins, or protein carrier transport. That's the term for it. But yeah, so something important to keep in mind is the rate of diffusion. We care a lot about the rate at which things enter and leave. And so remembering and understanding how these channels open and close and what they let in and out, that's very important in the medical, like having a medical aspect towards these things. And basically carried out by channels and carrier mediated transport. So what we're looking at here, we're going to have 3 different types of gated channels. These are very specific to certain things. And right here, this first one we're going to talk about is voltage gated. Voltage gated channels, hence the name voltage, purely depends on detecting voltage changes within the cell membrane. When a cell membrane detects a certain voltage change, this voltage gate, depending on what it's specific to, will open, allowing what's outside to come in. So it's purely based off of voltage. And when it reaches the mark that it needs, it's going to open, and when it's done, it closes. Mechanic gated channels are different because this idea of selectivity is very important here. A ligand or a protein is going to bind. It will come from the outside, and it will come in and it will bind onto a binding spot on this gate. And when this specific ligand attaches, this gate is going to flip open, and it's going to allow what needs to come in or out. It's purely dependent on a specific ligand that will come and it will bind and it will open. Kind of like a key. It only unlocks for that specific key. But this next idea of a channel, these aren't gated in the sense that they open and close. Leakage channels are constantly open. Leakage channels are constantly open. They don't close, but they are very specific. Very specific in terms of what they allow to pass through. So later we'll learn about a potassium leakage channel that helps dictate certain aspects about how our cell operates. I won't get into that, but it's important to remember constantly open, but very specific to what they let in. or out. This next idea is, so those are your gated channels. Next, we're going to talk about a carrier form of transport in and out of cells. It's still facilitated because it requires something to occur. But what facilitates this is this carrier protein will come, and the transport that happens is purely based upon the type of protein that this channel depends on. So again, it's very selective. Something to keep in mind with these is a great idea to have about them is they're more like a revolving door. It's only open and closed at one time. It's not a leakage channel, so it's not constantly open. But what will happen is a protein will come down, and this gate will open up towards this protein. And when that happens, this other side closes so that that can occur. And as this protein goes down, down the concentration gradient, this other side's going to open and release that protein on the inside or outside of the cell. And the idea of gradients is very important to remember because the concentration gradient of ions will always want to flow from high to low. Learn later that there's something called an electrical gradient as well, and those have to fight back and forth depending on what is most important. But all you need to know, protein carrier mediated transport, depending upon a protein, only open or closed at once. What can happen though is this idea of saturation. And that occurs when on either side we accrue a lot of these proteins, but because it only opens and closes either one at a time, what can happen is a point of saturation where there's a buildup of this ion or this chemical on one side of this membrane. And that's where this idea of the revolving door comes in. It can back up. It can get to a point where nothing can, things can get in, but it's like 50 people trying to walk through the same door at the same time. It just backs up. It's traffic. So three types of these carrier mediated channels is that we have these terms, uni, anti, and sim. When we're talking about this, we're talking about uniport, antiport, or simport. This just refers to the fact of, depending on what type of channel this is, this channel, a uniport will only allow one ion or chemical to go from its gradient, so from high to low. Uni meaning one. Anti is when you would have two proteins. So instead of one, or not proteins, chemicals, ions. So instead of only having one, you would have two. And one would want to flow in, so you would have, say, this green one. This green one would be wanting to flow out when this gate opens. And the blue one is wanting to flow in. So that's what we refer to as an antiport. Both are chasing their concentration gradient, trying to become equal on both sides, but they're happening against each other. Symport refers to symbiosis, together, and it just means going together. And this would refer to two of these ions. So forget the green one, but say we have this orange one now. this one would also want to flow in. And when we have two of these that are flowing in the same direction, that's what we refer to as a symport. So pretty straightforward. Just remember you have your gradients, concentration wanting to go from high to low. You have a rate of what's happening, and you have saturation. Those are really important things to take into consideration. So next we're going to talk about active transport, which is, we'll get to it. It's primary and secondary. It's two forms, but yeah. So this is diffusion. Next we'll talk about active transport. Okay, so these last two forms of transport are called active transport. Let me write that up here. Active. Both are active, and they're just two forms of it, so primary and secondary. Active refers to this need for energy to occur. So for primary, the most important, one of the most important pumps that we have that are classified under primary transport would be the sodium and potassium ATPase pumps. Primary active transport requires energy, and we use the energy in a form of ATP. So on this pump that we have, the sodium potassium ATPase pump, there's a binding site on the inside for ATP. And what happens is ATP comes in, gives one of the phosphates, becomes ADP, and that is the energy that's required for this pump to open. It's very important. Primary active transport moves two solutes against their gradients. You can see here, as the energy from ATP comes in, binds to the pump, 3 sodium is moved out while 2 potassium are moved in against their concentration gradients. This is done to maintain, again, this idea of homeostasis. And there's 2 conformational shifts that occur, and we refer to that as E1 and E2. This is just referring to the shifts in the form of that pump when ATP binds to it. So moves 2 solutes against their gradients, and this sodium-potassium pump is very important. It's probably a really good one to remember. So now moving to secondary. Secondary also moves solutes against its gradients, but the difference between secondary and primary, it would be that secondary doesn't require ATP. It uses energy that is stored in its concentration gradient, most times from sodium. So it works with a different form of energy to occur. We use terms symport and antiport for this as well when referring to the movement of these solutes against their concentration gradient. So it's important. The active transport moves things against their gradients, and they require energy. Diffusion, they didn't really have that. No energy is required for that. It's just kind of the kinetic energy that's naturally inside of our bodies that makes that occur. Honestly, that's the main difference between secondary and primary, is just the source of energy. And then, so it uses carrier proteins. And so the carrier proteins have a binding site on these pumps that move the solutes against their gradients and into the cell. And so allowing another solute to come in or out, anti-symport. That's what we're referring to here. So there's your 4 ways that, or 4 of the ways that we have things go in and out of our cells. Really important to just remember the distinctions between the active and your diffusion. So next, I'm going to finish by talking about osmolarity and tonicity, because when we have these things moving out and in of the cell, we're changing so much about how these cells react with our bodies, and the effect that this has on our physiology is just, it's phenomenal. So next we're going to talk about the effect that all these diffusions and modes of transport have on our bodies. So finish off by talking about that. Okay, so to finish up, we're going to be talking about osmolarity and tonicity. This time, instead of just having it drawn up, I'm going to draw as I go, because I think it'll help, maybe help the message make more sense. So the term osmolarity, I think a lot of, if you've taken chemistry, you've heard of molarity. Molarity is referring to the amount of moles that we have per liter of solution. Moles meaning, you have your mass and you have a molar mass of a chemical, and moles is the division between the two, and you'll get moles. So molarity, you have moles per liter solution. Osmolarity is talking about the amount of particles per liter of solution. So just a quick example, if we have, let's say, sodium chloride, very common. Sodium chloride is one molecule. So if we were talking about molarity, when we put sodium chloride inside of a solution, we would have 1 mole sodium chloride in that solution. But if we're talking osmolarity, sodium and chloride are 2 separate elements, 2 separate ions. So if they were put in a solution and separated and dissociated, we would end up with 2 different ions, sodium and chloride, and then that would give us a osmolar rate of 2 osmolars per liter of solution. So that's basically the idea. One, we're talking about a molecule, the moles per liter. The other, we're talking about the dissociated ions and particles within a solution. So that's what osmolarity is, and that's what we're going to be focusing on. So I drew 3 examples up here. I'll use them both for osmolarity and tonicity. Osmolarity, our cells have a default, we call it milliosmolar, amount of 300 milliosmolar in our body. This is just referring to a number associated with the homeostasis within those cells. Naturally rests at about 300 milliosmolar. So we'll say this is a cell. These are all 3 cells in our body. I drew it in beakers, so imagine it's in your body. Default, our cells have this milliosmolar level, which is 300. Each of these will have that. Milliosmolar. And I'll just say 300 again down here. So if that's the default level of the cells, when our body is going through homeostasis and fighting for equilibrium, those external factors and variables we talked about earlier that change the way our cells operate is going to have an effect on the cells themselves. And so us going into the medical field, we need to be able to understand what solutions can we put in the body to help the cells operate the way they need to. And so I'll write some terms up here. And first I'll talk about osmolarity. We have iso osmotic, hyper osmotic, and then hypo osmotic. And what these terms are referring to is the osmolarity of the cell compared to the solution, or the solution that we put into the extracellular fluid, which is what we talked about earlier. Intracellular fluid, extracellular fluid. Our extracellular fluid has two parts, the interstitial fluid and plasma. The interstitial fluid is the fluid that surrounds the cell. Plasma is just in the blood, the water in the blood. So when we say we put the cell in a solution, we're talking about putting a solution of dextrose or saline into the extracellular fluid. So, for example, here I drew two particles, okay? These particles, let's say, so two average things, or two common things that we put into IVs, which is intravenous fluids, that you'll see that at hospitals. You have a bag and it's attached to a vein that is entering into your blood, affecting your cells. And so, so two common things that we will use is dextrose, which is glucose, and saline. And we have figured out the amount to put in these IV bags to match the osmolarity of our cells. And that would be, you have a 5% dextrose solution, and you have a 0.9% saline solution. Okay, and it's important, these default numbers that we have, the 5%, 0.9%, these equal the 300 osmolar that our cells have. So if we were to use, say, a 2.5% dextrose, what that means is we're using half of 300. So these default have 300 to match the 300 inside of our cells. But what would happen if we put dextrose, let's say right here, because I drew 4 of these particles here to indicate 300. So if we put this dextrose into this solution right here, okay, and it also has a 300 milliosmolar solution, we need to know what the effect of dextrose is going to have on this cell. Okay, so two things. We need to remember permeability, and we need to remember what solutions our body metabolizes. Okay, so dextrose is permeable and we metabolize it. So the effect that dextrose is going to have when we put it into our extracellular fluid is it will go into this solution, okay? And that's where these terms are going to come into play. So if we put a 300 milliosmolar dextrose into the extracellular fluid, it matches the 300 milliosmolar in our cells. This is what we refer to as isoosmotic, iso meaning same. So when we put a 300 milliosmolar solution inside of our extracellular fluid, we refer to this as isoosmotic because both are the same. But this is where tonicity comes into play. Okay. Tonicity is the effect on the cell. Okay. So what effect would a isoosmotic solution that is permeable and metabolizes have on the cell itself? Okay. So just like these, isoosmotic, hyperosmotic, hypo, we're going to have isotonic hypertonic and hypotonic. Okay. So I'm gonna try to explain this best I can, as simply as I can. So we put a 300 milliosmolar solution of dextrose into this cell. We're going to refer to that as isoosmotic because we're matching the amount that's in the cell. But up here, I drew two particles to indicate half. So let's say instead of a 5% dextrose, which comes in at 300 milliosmolar, we're going to add a 2.5% dextrose solution. Okay? So now, if 5% is 300 milliosmolar, a 2.5% dextrose solution is going to be 150 milliosmolar. Okay? Because it's half. Amount is the same. We're not talking about half a liter now. We're referring to 1 liter with a different concentration of our dextrose. So if we put in 150 milliosmolar of dextrose, but the inside of the cell has 300, we're going to refer to that as hypoosmotic, meaning the concentration on the inside of the cell is more than the outside of the cell. So when we say it's a hypoosmotic solution, we're referring to the concentration of the solution that we put into the extracellular fluid. Now, over here, say we were to put a 6% dextrose, or let's say a 7.5% dextrose. Okay. Now, what we've done is we've put a solution into our extracellular fluid that has a higher concentration than that of the cell itself. So let's say now we have a solution with a 450 milliosmolar solution. Now we have a solution that is more osmolar on the outside than it is on the inside. So we're going to refer to this as a hyperosmotic, referring to more on the outside than on the inside. Now, what we've done here is distinguish the differences in osmolarity and our solutions. Now the idea is, what effect are these solutions going to have on the cell? Okay. So for this example, I've used dextrose. Dextrose is permeable to this membrane, which means it can flow in or out according to its concentration. And it metabolizes, meaning once it enters our cell is going to burn it. It's going to use it. That's what dextrose or glucose is this energy that our body's going to use. So, these terms, isotonic, hypertonic, hypotonic, what does it mean? Hypotonic is going to refer to a solution that makes the cell swell, grow, or another term is lyse. And what can happen is the bigger this cell swells, it can pop and it can just blow up. But hypertonic is going to refer to the fact that the cell is shrinking or shriveling because there's a higher concentration on the outside. Isotonic just means the solution that we put into the extracellular fluid is not going to have any effect on the cell. So tonicity is the effect that's going to happen. Osmolarity is referring to the flow of water of these solutions. Okay. let's have some examples of these terms and how we're going to apply it to these solutions that we've stated. So here I wrote down a 0.45% saline solution, and if this 0.9% is the 300 millismolar solution that we normally use, 0.45% is just going to be half of that. So we'll say that we put 150 millismolar solution of saline into the cell. So when we're talking about osmolarity and tonicity, what effect is this going to have on the cell? Well, saline is non-permeable, meaning that it cannot permeate through the cell membrane. So because we have more on the inside than on the outside, we're going to refer to that as hypoosmotic. But because the saline cannot get into the cell, we're going to keep the same concentration, so 300 and say 150. So tonicity-wise, because we still have more on the outside than on the inside, we're going to refer to this as a hypotonic solution as well. Because there's more still on the inside than on the outside. But let's look at this 5% solution of dextrose. Dextrose, we know, is permeable, but it's also metabolized in the cell. Glucose, dextrose, energy, that's the carbs that we eat and energy that we get through the food. So if we put a 5% dextrose solution into our extracellular fluid, surrounding a cell at 300 milliosmolar, what effect is this going to have? Well, a 5% dextrose is also 300 milliosmolar. So we'll start off isoosmotic because you have 300 on the inside or outside and then 300 on the inside. But because dextrose is permeable, it is going to flow into the cell. As it flows into the cell, it is also going to bring water with it. And so what's going to happen is this is going to continue to flow because it wants to create a harmony within its concentration gradient. So it'll keep moving until it creates, say, a 150, 150. But what happens is because it metabolizes, this dextrose is going to keep moving into the cell until it's completely gone. So even though we start off at isoosmotic, because water and this dextrose moved into the cell, now we have more on the inside than we do the outside. So what happens is we started at isoosmotic, but we ended at a hypotonic solution. Now, what would happen if we added both of them? So say we have this 5% dextrose and we have the 0.5% saline. So put in 450 milliosmolar, meaning the 300 default from our dextrose and the 150 from our 0.5% saline, because that is half of the normal solution that we use. So we put in 450. Osmolarity is going to tell us that there's more in the extracellular fluid than in the intracellular fluid, making this a hyperosmotic solution. This hyper part right here. What effect is this going to have on the cell though? That's what we need to think about. So because there's more on the inside and on the outside, well, what two solutions do we have here? So we have the dextrose, and then we have the saline. Well, the dextrose we know is permeable, and the saline we know is not permeable. Using the fact that we know that dextrose is also metabolized, what's going to happen is 300 milliosmolar solution of dextrose is going to move into our cell, okay? It's going to be metabolized just like we saw over here, and as it continues to flow, the cell is going to swell, and the ending that we're going to have erase this. We're going to end up with 150 milliosmolar saline on the outside of the cell. And so now our solution has more on the inside than on the outside. So that is what we're going to refer to as a hypotonic solution as well. Now, it's not drawn up here, but if we put a, let's say, if normal is 0.9%, let's say a 1%. If we put a 1% saline solution into a cell that's 300 milliosmolar, we know we have more of a milliosmolar saline than the 300 milliosmolar that we have in a cell. So say that this 450 milliosmolar is purely saline, okay? This will be a hyperosmotic solution as well, but the effect on the cell, because it's not permeable, we're going to end up with a hypertonic solution, meaning that we're going to have a higher concentration of saline on the outside than on the inside, but because it's non-permeable, there was no effect. So the concentration of saline on the outside is higher than on the inside, meaning that we have a hypertonic. So I hope these definitions make sense. Osmolarity, where is water moving? What is the solution that we have on the inside and outside? After water movement has taken place, if it takes place, what is the effect on the cell per solution, per type of solution that we're putting into it? So, yeah.